Hi, my name is Poom Suktitipat, and
I'm Young Park from Samsung Medical Center.
okay, I'm happy to introduce you to our first speaker, Dr. Ashita Mitra. I think all of you have heard her pitching this morning already. So briefly, Dr. Mitra is a group leader at the Telethon Kids Institute, Perth, Australia. Her lab is focused on understanding the early life interaction of human microbiome and immune system. She finished her PhD from the IISc in Bangalore and then moved on to did her postdoctoral training at the Singapore Immunology Network. Right? And then after that, she moved to Australia, and today we are very pleased to have her sharing her work with us on the topic human microbial immune priming in early life. Please welcome Dr. Mitra.
Thank you so much for the kind introduction. So I'm Archita, and I'm from Telethon Kids Institute. So my lab is interested in understanding the microbial immune priming in early life and how that shapes the lifelong immunity. And whenever you dive into the field of microbiome, one very pertinent question that comes to mind is how our body differentiates between the commensal microbes, which is our microbiome, and the pathogenic microbes. And to answer that question, I tried to answer this question in my PhD, in my postdoc in Singapore. And the way we approached this was to go much early into the development, which is the fetal development stage. And we asked this question that how early is too early? When do we encounter our first microbes? And when we do that, how our immune system corresponds to that or responds to that? So everybody knows that during birth, infancy, and toddlerhood, how important the fresh and good microbiome is for the developing child. But nobody knew what happens during gestation, and it was long believed that the womb is a sterile entity and there is no microbes present there. But we took up this question to answer whether there is any kind of microbial exposure in human tissues. So in Singapore Immunology Network with Flohan and our amazing clinical collaborator, Jerry, we started collecting the aborted fetuses samples. These were all clinical abortions, and these were all within second trimester range. And we divided our study into majorly 3 parts. The first part was to study the immune system, because that is important. We wanted to know how the immune system is developing and whether it shows any sign of antigenic exposure. The second part was to identify the microbes present in human tissues, the fetal tissues. And the third part was to recapitulate the system present within fetal tissues. So kind of putting the immune cells and microbes from the fetus together. So first I'll show you the first part where we employed the technology CyTOF to study the T cells in human fetal organs. And as you can see here, we found a lot of regulatory T cells in human fetal organs, especially in fetal gut and lung. And at the same time, we saw that these regulatory T cells, the suppressor T cells, they were high in PD-1 and Ki-67. So we thought there is some sort of antigenic exposure going on and active proliferation as well. And we went ahead and we tried to see what kind of antigenic exposure might be there, whether there is any sort of memory cells present there. And we indeed found, and this was very surprising for us, that there were a lot of memory T cells present in fetal organs, and they were effector memory T cells. They were showing TNF and IFN gamma expression. So we concluded that the fetal organs are experiencing some sort of antigenic exposure, and the memory cells are proliferating and they are responding towards those antigens. But what are those antigens? It could be anything, but we wanted to look at the microbial level and we wanted to see whether these are microbial antigens or not. So we went ahead and identified whether there is microbes present in fetal organs. So the species plot that you are seeing, it is basically the conclusion of 16S ribosomal RNA experiments, where we just take the bacterial RNA from each organ and we sort of try to amplify it and identify whether there is microbial diversity or not. And in this plot, you can see that as you go from peripheral organs, which is skin and placenta, to the internal organs like lung and gut, and to the very internal organ, which is like thymus and spleen, which could be positive control, internal positive control, the diversity of the microbiome decreases, which means the fetal organs which are exposed, like skin, placenta, lungs, and gut, they had high diversity in microbial species or genera. And we wanted to confirm this and wanted to remove all the confounding factors like contamination or batch effect. We sent some samples to our collaborators in Weizmann Institute, Ravid Strassman's lab, and they also found the similar results. So the diversity was present there in fetal tissues. And at the same time, we wanted to study the gut specifically because we know a great deal about gut microbiome. So we wanted to see what is happening in the fetal gut. And here you can see we found more than 11 specific genera which were specifically enriched in fetal gut, and they were not present in either external control, which is PBS, or the internal control, which is spleen. But all these were DNA and nucleic acid. We wanted to see whether these microbes were actually alive, or is this something just flowing through the placenta through certain cellular carriers. So we went ahead and we cultured these bacteria, and we were able to culture most of them, and to our interest, they were mostly anaerobic or facultative, which might be the case with fetal tissues because the fetal environment is not very rich in oxygen, we know now. But all this was again just culturing and sequencing. We wanted to see whether there is bacteria present or not. So we took the help of our amazing collaborator, Benoit, and we went ahead and did the scanning electron microscopy. And we found the bacteria-like structures present within the fetal intestine, which you see here on the left-hand side. I'm not sure if the pointer... Okay, sorry, I don't know. On the red dots show you the bacteria which is present, which were like a circular bacteria-like structure. And on the right-hand side, you are looking at the regions where there was no bacteria. So it wasn't like there was bacteria present everywhere. It was very highly speckled across certain regions, and those regions had mucin-like structures as well, which we know from adult biology that the microbiota present in intestine is mostly associated with mucin-like threads. So we found that, and we did not find any bacteria in 10-week or 9-week-old fetuses. And but every technology has its limitation. This was again only just going by the structural prediction. So we took help of our other colleagues, Dr. Ankur Sharma and Riya Pai, and we did RNA scope analysis, which is basically to probe the bacterial RNA within the tissues. And we found that the bacterial RNA was present. So the green color on the first one is the bacterial RNA, and it was very nicely aligned across the epithelium. You can see the EpCAM marker as well, and merged. And here the fetal liver or fetal lymph node didn't have any microbes present. So by this we knew that there is microbe, it is alive, and we could see it also. But we wanted to recapitulate the fetal system and see what they are doing with the immune system, because earlier we saw that there are memory cells present in fetal tissues. So we recapitulated a system where we took, so it is a very syngenic system where we took the bacteria, the DCs and the T cells from the same fetal gut. And then we tried to first expose the DCs with these bacteria so they can take up the bacteria, and then they present the bacteria to the T cells. And we wanted to see whether these T cells have any memory against these bacteria. And we were very glad to see that there was memory against these bacteria. Here in the right-hand side plot, you can see in the lower two panel, the orange and the blue, there was a shift. There was a very nice shift when we presented the bacteria to the T cells. And we did it with fixed and non-fixed DCs just to see whether the exposure is antigenic mediated or the cytokine mediated. We found the expansion in both the cases, which means the exposure is mediated by both antigenic and the cytokine response as well. But when we do the fetal staph, which is a fetal bacteria, DC with PFA, where we are basically blocking all kind of cytokine production, the level of expansion was highly significant, which means the antigenic exposure was significant enough to elicit memory response in fetal tissues. And these memory cells were effector memory in nature. So it kind of summed up the entire study, where we could show that the fetal organs have microbes, they have memory against these microbes, and this memory is not just latent memory, it is effector memory. We still don't know what it does exactly and where it resides. We know that it resides in the fetal lymph node, but we don't know like what time it comes, and that is a question of active exploration. But at the same time, we wanted to see where the bacteria is literally interacting with these immune cells. So in my lab, I'm developing this model where we are looking at the microbiome using this the RNA-scope technology, which I just showed you, and the immune system of the host using spatial transcriptomics, and combining them together and looking at looking at the microbes and the immune system at spatial level. Combining them together. So this will give us the information when and where the bacteria interacts with the immune cell, and when it does, what kind of response is generated. So we are currently doing this in fetal and neonatal system. So I'll just give you an example here with the initial data that we have got. On the left hand, so it's both the same plot, but only the markers on the top has changed. So I'll explain you. On the left hand side, you can see there are certain regions which are in blue that are non-immune rich regions. So within the fetal intestine, there are some regions where there are no immune cells, which is quite intuitive for a fetal system. But then there were clusters which were highly rich in immune cells, which is the pink one. And within that cluster, there are different kinds of immunity present. So as you can see, within that, the cluster number 2 and 3 differ by the expression of TBET and CTLA4. So there were some clusters which were high in TBET and CTLA4 expressing regulatory T cells. And the same regions, when you see here at the extreme right, cluster number 3, were high in effector memory T cells as well. So it was kind of very intuitive to see that wherever the suppression is happening, the more suppression is happening, you will see not many effector memory T cells. But when the suppression is not so pronounced and you have a Th1 kind of response, then we have effector memory T cells coming there. And we are exploring this more in detail, like what these regions are and whether there is any bacteria present in this region. That's what we are currently developing in the lab. And here, another thing, because why we are asking these questions is that whenever a baby is born preterm, they are highly vulnerable to infections, and they encounter infections by those bacteria which are otherwise harmless. And one such infectious model is necrotizing enterocolitis, which is highly debilitating and often fatal. So we are taking the tissue biopsies from these babies which are undergoing treatment for necrotizing enterocolitis, and all these babies are hardly 24 to 28 weeks old, and they are born premature. And we are looking at the immune profile and the bacterial profile of these babies' gut. And as you can see on the left-hand side, so this is the spatial transcriptomics data. And on the left-hand side, you can see a lot of inflammation happening in this gut, which is identified by CD45 and CD3 immune cells. And on the right-hand side, this portion of gut does not have any inflammation going on, not so much. So this is the initial results from the lab. And we are now further exploring like what kind of immune cells are associated with highly inflamed regions, and whether there is any specific bacteria present or absent there, which should have been present there in order for the system to cooperate with the external bacteria. So the whole idea is that how our system differentiates between commensal and pathogenic, and we believe that this priming happens much early in development, and there are certain bacteria and certain type of immune cells which come together, and they impart this kind of immunity to the baby to face the external world. So the overall aim of the lab is to understand the existence and identity of microbiome, especially in early life, then to understand how they prime the immune system, so what are the key immune cells important for this feat, and then how it can help to protect the child against further infections, autoimmunity, allergy. So we are now taking up cohorts which we can follow for next few years to see whether they develop any kind of allergy or autoimmunity or any other kind of conditions. So the Mishra lab is all set up now, and we are looking for more motivated PhD postdoc fellows to join us. We are hiring. So please write to me if you are interested, and I'll conclude with that. Thank you.
Thank you for a very eye-opening talk. Any, please, questions?
I have some question. So in mice, it's been known for a while that gut Th17 cells are dependent on microbiota, right? And it's been discovered that segmentus filamentous bacteria is driving gut Th17 cells development in mice, but there's no such bacteria in human. Yeah. So have you looked more into gut Th17 cell, and do you think it's more likely than not that you will discover something that induce human gut Th17 cells?
Yeah, that's very interesting question. And we are actually, I didn't show that data today because only 10 minutes. We have some single cell data from human fetal organs across like the lymph node, gut, and lung. And we find Th17 positive cells in the lymph nodes for the gut, but we don't find it in spleen or thymus or any other organs. So it's perhaps doing something for the microbiota. Again, I would say that the mice system and the human system is quite different when it comes to microbiota, because the diversity, it's not present there in mice one. The other thing is how it interacts with the immune cells. It's very different. So it will be interesting to see whether it is the same kind of cells like CD03 positive cells and Th17 cells, or is it something different altogether which we discover.
Thank you.
Yeah. Just have a sort of practical question. How frequent were these memory cells, like in tissues like the lung, in your hands?
Yeah, so if I understand it correctly, so we found throughout second trimester of gestation, we found that there were memory T cells present in these organs. Whether it is a snapshot in time and whether they are present there for long-term memories.
So you can detect them, but are they very rare, or I didn't catch it on your slides?
Okay, sorry. Yeah, so they were not rare actually, but yeah, it's only 5%, 3 to 5%. So yeah, it was more in gut than lung, of course, but yeah.
Okay, maybe one last question.
Yeah. Great talk, thank you. I'm Yoshi from Osaka University. So I have a question about the bacterial component of fetal tissue. So did you observe the tissue-specific pattern of genus or species level composition of bacteria? I mean the bacterial death or prebacterial environment in gut tissue or something like that.
So if I got it correct, you're asking whether these bacteria were actually integrated in the gut tissues.
No, my question is about the composition of the each tissue microbiome is consistent to the pattern.
Okay. Yeah, so it's very interesting question. Actually, we did find some diversity among organs, definitely, and we also found diversity among fetal samples. And that's why we did not go into the exact identification of the bacteria. We did not go to species level. We only went till genera level because it's sometimes very difficult to identify it with 16S ribosomal RNA sequencing. But yeah, we did find different bacteria present in different organs, and sometimes they were different within fetuses as well. And I think for that, we need more comprehensive approach and more sample size to understand, because again, the samples that we get, they might be coming from different ethnicities, different stages of gestation, and maybe mother is having antibiotic or not having antibiotics. So all that information was not present in the earlier data set. Now we are including all those information as well. So yeah, there was diversity, to answer your question.
Thank you.
Yeah.
All right, please join me to thank Dr. Mishra again.
Thank you.
Okay, for our next speaker is Professor Manop Pitakpakorn. Okay, Professor Manop is a professor of medicine and a clinical staff at Faculty of Medicine, Sirat Hospital, Mahidol University, Thailand. Right, so he's actually one of the pioneer of genomics medicine and precision medicine in Thailand. Among many of his accomplishments, I think he's the one who helped Thailand move forward in terms of cancer precision medicine. And we are hoping that, I think now, since he's with us today, he would also help us and Thailand to, I think, adopt some of the results from single cell studies and apply to precision medicine as well. And so today he's going to share with us his thoughts on how we might be able to use the transcriptome data from single cell sequencing towards the application of precision medicine. Please welcome Dr. Man.
Good afternoon, everyone. Thank you for kind introductions. This topic is going to be totally different view, compared to the other presentation in the past 2 days. And I consider myself an outsider of the communities because I'm actually a clinician practicing and seeing patients. I'm practicing mainly in clinical genetics and also running a clinical genomics lab for the hospital. So majority of my work on the clinical part of the various kind of genetic disorders, mostly monogenic, like from rare diseases into more common one like cancer. So this one is something that in genomic community greatly need. Which is a reference kinds of data in transcriptome level. I think would help a lot in terms of the clinical medicine. I think people truly realize that the great benefit of the Human Genome Project back about 30 years ago. Right now, genomic medicine become, I would say, mainstream, and we use a widespread adoption of genome technology like genome sequencing in terms of diagnosis, treatment selection, and also prevention of certain diseases. But moving from research to clinical diagnosis in the past 30 years, have a lot of challenges. It doesn't mean that technology is not great, but moving from research perspective into the full clinical services have a lot of challenges that we have to overcome. For example, standardization, information need to be highly accurate. And accurate enough for clinicians to make clinical decisions and also being informative to both patients and clinicians. So even 30 years has passed, many barriers still exist, and I'm going to show you just some example. I think one of the great benefits that we can fully adopt genome sequencing into the clinical medicine is because we have a human genome reference from Human Genome Project. In the past 20 years, from the first draft of Human Genome to the current one, we have like 38 editions of reference genome now with several hundreds of minor revisions within that. And we just recently have complete telomere to telomere human genome sequence this year. And with this gives rise to various databases. For example, variation SNP database, a healthy population database, or even large genome database like NOMAD that we use the power of the diversity and allele frequency to determine the pathogenicity of variants. But with that, we still face a lot of challenge from the genetic variants that we identify from genome sequencing. How do we determine that variants to be pathogenic? At least we have to go through the standard procedure like computational models, evidences, segregation analysis, or large population database. But to confirm this, ideally we need the functional studies. Obviously, It's entirely impossible for every single gene at the moment. We don't have the animal models for every single genes. It's almost impossible to have a human model, even though there are some natural occurring human models that we can study. Some people might try to develop cell models, but it's still a lot of challenge that face us. A lot of genes that we still do not understand the functions. Those are the gaps that we need to fill. Once we do genome sequencing for the known or common variants in common genes, it's not difficult to make clinical decision based on the results. But for the novel or rare variants, how do we interpret it? That's identify variants to be pathogenic versus benign. There are frameworks that we are using in the communities, like we look at the functional consequences. Does this variant impact the protein function or gene functions, being loss of function by computational tools, something like that? Pathogenic assessment, can we impact that impact of variants leads to disease phenotype or not? Or at the individual level, does those genetic variants cause symptoms in those patients? So I think many people are familiar with this 2x2 matrix, that we have a gene level of causation and variant level of evidence. For the gene level, if there are known genes, it's easy, like on the right side of the matrix, either have a large impact being pathogenic, or they have unknown impact that we consider as a VUS. But for the genes that we do not understand, it's very difficult to determine the causation or how to interpret it. Since the abundance of genome sequencing that we use in research and clinical services, a majority of the newly discovered genes are found first by sequencing. And these are totally different from the past, that we observe the clinical phenotype first, the abnormality that in the animal model or disease models. We dig down into the protein level, RNA level, gene level, and then that's easier to understand the function of the gene. But the newer discovered genes are difficult to interpret. What's the meaning of it? And this also create a lot of headache when we do sequencing, because in undiagnosed or rare diseases, majority of the variants identified in patients are private mutations. We cannot see in other family or published data. How do we interpret from the information that we have? And there are a lot of misdiagnoses in the past. Even before we have more knowledge about large database and ethnic diversity, like Jay or Chaim has stressed in the first day. One of the great examples, and we have learned a lesson, is the cohort of hypertrophic cardiomyopathy, that in the past, there are several genes that have been discovered, some pathogenic variants have been published, and we diagnose a lot of patients and mistakenly diagnose them with hypertrophic cardiomyopathy, especially in African American patients, because it is later found that some variants are common in that ethnic groups. And it's not truly pathogenic, and this has resulted in several hundred patients. It's been unnecessary implant with portable cardioverter defibrillator, just being diagnosed with this pathogenic variant that is not truly pathogenic. And there are other examples in the newly discovered data, like a PROMISE cohort, that is the cohort of South Asian descent that live in UK, and they have some degree of consanguinity, and we can use these natural occurring human knockout models, because some normal people that do have what we call homozygous loss of function variants in some genes, and we found that around 5 to 6% of human genes are truly non-functional in this population without any health impact. What it means that about 5% of human gene, we totally don't need it. If it's totally deleted, we are still fine. So if we found those, how do we make a meaning in terms of clinical interpretation? So at the moment, we use widely adopt ACMG standard classification of pathogenic variants. The variant interpretation are mainly for clinical use, but even we have a stringent criteria, a lot of domain we still don't have enough data, especially for the functional one. So we are mainly relying on either population data, computational data analysis, and functional data in some sort, like the data on the RNA. In silico prediction, I think for the people who runs this algorithm and use a lot, still familiar, we still have a lot of algorithm that's being developed. In clinical labs, we normally rely on between 5 to 12 prediction models to help us interpret this. Why don't we use just one or two? Because prediction is prediction. We still cannot make a final conclusion on it. It's similar to probably the weather forecast model, something like that. And even the in silico prediction, there's still some limitation we're still facing a lot. For example, silent variants, how do we know it's not creating novel splice site? Computational model prediction is not highly accurate with this. Initiation site variants like start codon variants are not always deleterious because there are always alternative initiation sites in some genes. The relevance to disease pathogenicity, we don't know the expression level of these genes in different tissue or even organs. The organ, does the level of expression correspond with the disease phenotype or not? Something like that. And with the clinical genome lab, we still face a lot of problematic variant interpretation. We need to finally resolve with the functional interpretation or functional study. These are just some of them that we see all the time, like cancer genes. The in silico prediction predict as deleterious variant, like nonsense variants, splicing variants. But actually, the functional study are showing as benign. Those are benign preserved functions, or even silent synonymous variants like TP53, the last one. When you see this, it's simply benign, right, tolerate variant, but it's actually pathogenic because in functional study, we found that the variant induced outer splicing, which result in disrupted peptide, and there are several groups that also show this. So how do we make out this? With the clinical adoption, we see sequencing or genome testing become a standard of care, like cancer. These are the data from Thai cancer patients, over 4,000 patients. Even though we identify 1 in 6 patients being harboring pathogenic variants, but about 35% we still don't know that variant, what it means. And the implication of this create a lot of uncertainty, anxiety, both on patient side and clinicians. Several labs trying to adopt the RNA studies just to resolve some of the issue on the VUS. These are just one example that the RNA data can resolve a lot of uncertainty in the VUS area. And we hope that these are just one of the issue for the consideration for the communities that, even though genomic medicine has come a long way, and we think that the transcriptome data will take a lot of the medical impact and meaningful impact in the future, but we would still face a lot of things that we need to be prepared. And reference transcriptome, I believe that would be a great tools to resolve all those uncertainty, especially in the widespread adoption in clinical medicine. Thank you.
Any questions from the floor? Dr. Varodam.
Right, thanks so much for a great talk. Probably just quick questions. So I think for those who don't yet know Ajahn, Professor Manop is also a very influential, right, speakers in terms of dealing with public and also policymakers. So I think we pretty much convinced already, right, that having single cell transcriptomics and so on and so forth, right, is important to get us to the next level and so on. But my question is that because Human Genome Project has been around for a long time, right, and it's pretty well established, right. I mean, among I guess public that this is something important, and they kind of have the impressions that this is the one that's going to solve everything. But now we're telling them already, right? Okay, that's not the end, right? We have a lot more to go into, right? So I just wonder, from your point of view, right, in terms of influencer, how do we keep them interested, if that makes sense, and basically, still get support and funding in the next chapter, if that makes sense.
It's interesting questions, probably not entirely relevant to the community at the moment, but I believe for the whole scientific communities, I think we face the same problems. We are exciting in new discovery, high technology, bringing new knowledge to the world, but as an outsider, like public policymakers, they don't really fully understand, and the majority of them are still seeing doing research as a great expense, and they don't see any substantial return, something like that. One is I would go with something that we could adopt in the short period of time. For example, I think COVID situation has give the great examples to the policymaker how science have a lot of potential in terms of solving those crises. The SCA data provides the insight into SARS-CoV-2 viral entry and ACE expression, something like that, that would keep public appreciate hard work from our community, I think.
Here. I think thank you for telling us that there is a large gap between the research tools and the clinical utility. The 4,000 odd patients that you showed, are they all non-ovarian, non-breast, or they are all ovarian and breast?
Yes.
Say it again, sorry?
The 4,000 patient data that you showed, are they only ovarian and breast hereditary cancer, or non-ovarian, non-breast?
Or there are a variety of cancer, like ovarian cancer, breast cancer, colorectal cancer, pancreatic cancer, endometrial cancer, or even the people who have multiple cancer but doesn't fit into a common one. Yeah, we group together. We do the sequencing for everyone who have a clinical suspicion of hereditary cancer.
Okay, thank you.
Maybe one last short question.
Okay, maybe not me then. So I was wondering if there is a role for things like deep saturation mutagenesis scans, or these sort of assays that really are used to dissect an entire element in a sort of population agnostic way in closing this gap in VUSs, and whether you think they're suitable or they're too removed from the actual cancer phenotype and the cancer cell?
Great question. Actually, it's one of the great tools, especially when we have a lot of the easy tool for genome editing, like CRISPR-based mutagenesis. But one of the problem is a lot of genes we still don't know how to monitor the phenotype based on the mutagenesis we create. It's become a very, I would say, difficult to see the effect of those mutation that we create. And those are very useful in the known function genes, but for the unknown one, I think the best that we could do is to look at the transcriptome level, just to make sure that how those variants impact the structure or the transcription.
Thank you.
Okay, so please join me to thank Professor Manop again. Okay, our next speaker is Professor Yosuke Togashi. He will be joining with us online. Professor Togashi is a professor in Okayama University. He finished his MD from Kyoto University School of Medicine and received his PhD from Kindai University. So today he's going to share with us talk about clonal evolution in tumor microenvironment.
Thank you, Chair and the Program Committee for giving me this opportunity. I'm Yosuke Togashi from Okayama University. I'm going to talk about clonal evolution in the tumor microenvironment. I focused on cancer immunology, especially tumor microenvironment, using clinical sample. Tumor tissue are obtained from patient, which was digested into single-cell suspension. Afterward, we analyze TIL using flow cytometry, mass cytometry, or single-cell sequencing. We worked on biomarker study using clinical sample. About 100 patient clinical sample who received PD-1 blockade therapy, and we analyzed TIL using flow cytometry and mass cytometry. With top 2 panel responder, bottom 2 panel non-responder, we focused on this cluster. This cluster was dominant in responder and not dominant in non-responder. This cluster was PD-1 positive CD8 positive effector T cell. So PD-1 positive CD8 positive effector T cell could be a predictive biomarker, but not perfect biomarker using 100 patient clinical sample. So next step, we use single-cell sequencing, especially to elucidate comprehensive phenotype according to each T-cell chronotype. We perform single-cell RNA and TCR sequencing for melanoma, for TIL to PBL. This is a simple UMAP cluster figure. 6 samples were merged, one UMAP figure. 1, 2, 3, 4, 5, 6, 7, 8. The T cells were clustered into 8 clusters, and we focused on this purple cluster because this purple cluster had high PD-1, TIM3, LAG3, CTLA4 expression. These molecules are so-called exhaustion-related molecules, so we named this cluster exhausted cluster, TEX cluster. As the TCR, we calculate the TCR diversity score. The text cluster had highly skewed T-cell chronotype. However, when top 10 chronotypes were plotted into each patient UMAP figure, other cluster, not text cluster, had also skewed chronotype. So to search for cancer-specific T-cell, cancer-specific T-cell mean T-cell directly attack cancer cells. Cancer-specific T-cell, we selected top 20 chronotype from various clusters, of course including text cluster, but not only text cluster, but also other cluster. For example, number 1 from non-text cluster, number 2 from text cluster. We created TCR T cells using each TCR sequences. And the TCR T cells were co-cultured with autologous cancer cells and were analyzed with flow cytometry. As a result, number 1 cluster, number 1 chronotype from non-text cluster did not respond. Number 2 chronotype from text cluster clearly responded to autologous cancer cells. Other chronotype showed similar tendency. So cancer-specific T cell clone had exhaustion phenotype. From these gene expression data, we identified several specific molecules in the cancer-specific text cluster. Among them, we focused one molecule because we created conditional knockout mouse, CD8 positive T cell specific conditional knockout mouse. In this mouse, tumor growth was very slow, and PD-1 blockade therapy exhibit dramatic efficacy. So this molecule could be a novel therapeutic target. We analyzed both pre and post-treatment TIL from a super responder to PD-1 blockade therapy. Paired TIL and we obtained paired TIL sample. Blue bar mean pre-treatment, orange bar mean post-treatment. There are dynamic change in several clusters, in various clusters. At first, we focus on text cluster, of course text cluster, cancer-specific text cluster. Cancer-specific text cluster increased after the treatment. Next, we merged chronotype, pre-treatment chronotype and post-treatment chronotype, purple mean text chronotype. For example, these two chronotype, pre and post, considerably existing chronotype, contracted text chronotype. In contrast, these chronotype expand after the treatment, and these chronotype newly infiltrated chronotype. So PD-1 blockade therapy promote novel cancer-specific text chronotype infiltration. However, such text chronotype were rarely found in peripheral blood. So next, our question is, where do such chronotypes come from? There are many data, but to make a long story short, we next focused on lymph node, non-metastatic lymph node. So we collected paired sample PBL, non-metastatic draining lymph node and TIL. And single cell TCR RNA and TCR seq were performed. As a result, connected line means same chronotype and purple mean text chronotype. As you can see here, many chronotype, text chronotype were shared between TIL and RNA. However, rare chronotype, text chronotype were shared between TIL and PBL or RNA and PBL. These data suggest the importance of cancer-specific text chronotype from lymph node. So we validate these data using a mouse model. We use sensitive mouse cancer cell line to PD-1 blockade and analyze TIL, especially PD-1 positive effector T cell. After PD-1 treatment, PD-1 blockade, PD-1 positive effector T cell increased in the TME. However, when we use FTY720 homing inhibitor from lymph node, cancelled the increase completely. In contrast, in a resistant mouse model, B16F10 mouse model, the increase was not observed after PD-1 blockade. In addition, sensitive mouse model, FTY720 homing inhibitor completely cancelled the PD-1 blockade mediated efficacy. So these data suggest the importance of cancer-specific text infiltration from draining lymph node. This is summary. Cancer-specific T cell chronotype had exhaustion phenotype. Such chronotype was rarely found in peripheral blood. PD-1 blockade therapy PD-1 blockade therapy promote expansion of such chronotype and novel infiltration such chronotype from draining lymph node. Back to this slide, there are other cluster, increasing cluster after PD-1 blockade therapy. We next focused on follicular helper T cell. Follicular helper T cell, originally PD-1, follicular helper T cell had originally high PD-1 expression, so PD-1 blockade increased follicular helper T cell is very reasonable data. In addition to PD-1 expression, we found CXCL13, which reportedly recruit follicular helper T cell, but CXCL13 was highly expressed in cancer-specific text cluster. PD-1 blockade therapy increased this expression, especially in cancer-specific text cluster. In mouse model, CD8 positive T cell depletion cancels the infiltration of follicular helper T cell. So CD8 positive T cell, especially cancer-specific text, which has high CXCL13 expression, play important role in TFH infiltration. In addition, PD-1 blockade increased follicular helper T cell infiltration, as was observed in human clinical sample. And follicular helper T cell knockout mouse, PD-1 blockade mediated efficacy was impaired in knockout mouse. So, follicular helper T cell in the tumor microenvironment was important for anti-tumor immunity. In addition, we further classify the tumor-infiltrating follicular helper T cell into two clusters, orange and blue cluster. As the TCR, considerable chronotype was shared between the two clusters, suggesting continuous differentiation between the two clusters. We are now investigating the functional difference between these two clusters, and we of course analyze RNA velocity or diffusion map pseudo time analysis. Final slide. I appreciate all collaborators, clinical department, and data analysis facility. Thank you for your attention.
Due to limited time that we have, so we might have to skip the question and answer.
Thank you, Dr. Tokashin. We're gonna close the talk. Thank you.
Thank you.
Thank you.
So we have limited time. So we have 3 speakers in about 20 minutes. So sorry, but so we're gonna ask the speakers to finish that in 10 minutes, and then yeah. Next speaker will be Dr. Hong Seung-hee from Yonsei University in Korea. Actually, she was trained in the Case Western. I also was working in the Cornell Medical School, and then she's expert in the lupus and autoimmune diseases. So she's gonna present us today about autoimmune disease.
Yeah, thank you for the nice introduction, and thank you for inviting me to this awesome conference. So today's talk, I'm gonna briefly talk about what I have done before I had my own lab while I was working in US. And I started my own lab about a year ago. So I'm gonna present unpublished data that I have generated within a year. So my main interest is about autoimmune disease. So autoimmune disease, as you know, it's a condition that host immune system attacks its own host. And it is known that on the genetic predisposition, when environmental factor comes, immune dysregulation will be started and autoimmune disease will be manifested. And the exact cause of autoimmune disease is not known yet, and it's it's very complex, and many different types of immune cells are known to be playing a role in a different way for each autoimmune disease. But for some autoimmune disease, the main players are revealed. For example, for atopic dermatitis, Th2 and Treg cells are known to be playing an important role. And for the psoriasis, Th17 is dysregulated. But for most of the autoimmune disease, all types of different kind of immune cells are intermingled in the pathogenesis of the disease. And for example, lupus, I think it's one of the most heterogeneous disease. And there are so many different immune subsets has been revealed to playing a role in the pathogenesis. And there is a huge heterogeneity within the disease on the subject level. So I have been working on lupus for the last 10 years, and its main characteristics is a clinical heterogeneity, and it can affect everywhere. And there is no predictors for end organ involvement, and steroids continue to be the first line of therapy. But many clinical trials targeting the lupus has been failed, and only 2 drugs has been approved in like last 60 years. And the challenge for the lupus research is coming from highly unpredictable disease courses, as you can see. And disease activities keep changing, and the patient experience continuous flare and remission and flare and remission. And what I did was I did personalized immunomonitoring to uncover immune networks that correlate with the patient's disease activity changes. So I used microarray data, the longitudinal data, and I tried to find the immune networks that is correlating with the disease activity changes for every patient. And then I could stratify the SLE patient into the 7 groups. And I found these 5 immune networks are correlating with the disease activity in a different way for each patient group, so that we could see that if we want to use B-cell targeted therapy, this group of patients will be more promising because they showed great correlation of their plasma blast signature with the disease activity. But for the interferon blockade, this group of patients, different group of patients will be responsive because they show interferon signature correlation with the disease activity. So this kind of analysis gave us clue about the heterogeneity of the disease, and it was further studied with the single-cell RNA sequencing analysis with a higher resolution. And it has been published 2 years back. So I'm not going to go on detail about this project today because it's already published. But I want to switch a gear to unpublished data that I have generated in my lab currently. So especially I'm going to talk about atopic dermatitis result. So atopic dermatitis is a chronic skin inflammation condition, and patients suffer from continuous itchiness and scratches. And its main player is known to be a Th2 cell, and targeting this Th2 cell using IL-13 or IL-4 blocker has been pretty effective. And but there are many different types of immune cells are also involved in the pathogenesis of the disease, and the systemic immune networks in the pathogenesis of this atopic dermatitis has not been clearly understood yet. So to answer that question, we did PBMC single-cell RNA sequencing analysis using 12 AD patients and 6 healthy control. And we purposely recruited only severe patients. And this severe patient, we had even a sicker patient, so that we classified the patient into severe and the very severe patient. And all these patients were treatment naive. We did clustering and annotation, and we found some clusters were significantly expanded in AD, including CD4 memory T cell, monocyte, and proliferating T cell. And the expansion of the memory T cell was kind of expected because of this Th2 and other types of T cell dysregulation. So we try to look at the Th2 and Treg inside the memory T cell, and we did transcription factor prediction analysis using PyCynic, and we could locate the Th2 and Treg, and we found this AD patient shows expanded Th2 and Treg activation. And also, we found a decreased cluster in AD, including NK cells, PDC, and CDCs. And we looked at the T cell more closely by comparing AD versus healthy or the AD expanded T cell. And we found AD patient shows upregulation of CCR4 and CCR10, which is indicative of Th22 signature. And also we found downregulation of cytotoxicity-related genes. So the decreased cytotoxicity in AD has been reported in some mouse model and bulk RNA-seq research, but this was kind of first report that was found in the single-cell RNA sequencing data. And we also looked at the NK cell more closely, and we found 4 NK subset, and all this NK subset was decreased in number in AD patient. And we also looked at their gene expression profile by comparing healthy severe AD and very severe AD patient, and we found this cytotoxicity molecule expression was highly downregulated in the very severe AD patient, and also interferon signature was increased. So the AD displays a decreased number of NK cells, which showed effective cytotoxic program. And we also looked at myeloid cells, and myeloid cells showed very specific 3 clusters that was highly expanded in very severe AD patient, and they showed increased innate immunity and TLR-related marker in very severe patient, and also type 1 interferon. So this analysis result indicate the involvement of innate immunity-related pathways, including NK cell and myeloid cell. And I didn't show everything today, but this is in other cells in terms of pathogenesis of the atopic dermatitis. Finally, I tried to stratify all the patients in healthy control using the single-cell subset proportion. And it was striking to see the clear distinction between the healthy cluster and the AD clusters. So all the healthy cluster showed higher proportion of cytotoxicity-related cluster, like NK cell, cytotoxic, CDC, and PDC, and etc. And we did pathway analysis, and we found the cytotoxicity-related gene was also highly enriched in the in the healthy control subset. But in the left-hand side, we found AD-specific cluster, and we also found some very severe patient were clustered separately from the other AD patient, and they showed higher proportion of this specific cluster, which include ISG high monocyte, ILC2, and specific TC and specific B cell. And we also found interferon-stimulated gene expression was high, and the TLR signature and Nachi signaling was also unregulated in this cluster. So this shows us the basis for the therapeutic target for specific AD patient. So I want to acknowledge my previous PI, Virginia Pasquale, who I have been working together for 10 years for lupus research mostly. And I made this fantastic new collaboration after I started my own lab in Korea. Donghoon recruited the atopic dermatitis patient, and Hyunjae generated the data, and Sunpil gave me a lot of clinical implication. And those, all these guys are dermatologists, and we had a fantastic collaboration so far. And I also want to thank to all my students here, especially Kyungcheon Lee, who had analyzed the data and generated all the figures, and all these other students in my lab. Thank you. Thank you for listening.
Thank you, Dr. Hong. So we limit. Maybe if you have any questions after the session, you can ask her directly. So next speaker will be Dr. In-Kyung Jung from KAIST, Korea. So he working on the epigenetic regulation of the human diseases. Then our present our state today about the brain disease.
Okay, so thank you for the introduction. And let me see. Okay, so I'm In-Kyung, and my lab is mainly working on the epigenetic gene regulation in various complex human disease. And today I'm very glad to share one of our recent work about the Parkinson's disease. Okay, so as you know, the Parkinson's disease is the second most prevalent neurodegenerative disease in the world. And pathologically, the Parkinson's disease can be characterized by the loss of the dopamine neurons. However, for individual the patient, each patient subject to various both motor and non-motor symptoms, and also they might be involved in various complex molecular mechanisms. So in order to understand such complex molecular mechanisms underlying the Parkinson's disease, there are a lot of the studies have been made to identify genetic risk factors. And thanks to the power of the genome-wide association studies, we can identify over 3,000 important variants presented here. However, we still fail to provide the detailed mechanism of this variant, because as you can see here, all these variants are mostly located in the non-coding areas. So this is a very difficult problem to define their exact function. So in order to annotate their function, we hypothesize that these non-coding risk variant can contribute to the disease by perturbing sequence of the regulatory element in the genome. And thereby, they can cause the dysregulation of the gene expression in a specific cell type. Test this hypothesis, however, is limited by two important the limitations. The first, actually the annotation of this regulatory element is not complete in a cell type specific manner. And also, it is very difficult to define the target gene of this regulatory sequence, because many of these regulatory sequences can control target gene expression over large genomic distance. So in order to address these two main hurdles, we proposed a single-cell multi-omic approaches with the patient brain tissues. So for this end, we first collect the patient brain tissues and also age-matched normal tissues. And we also provide the single-cell, the epigenome and transcriptome using these tissue samples. And by collecting the previously reported important genetic variant, and also by generating the high-resolution chromatin contact information by using the same tissue that we collected from this study, we try to define the target gene, the information. So by using this variable resource, we first try to identify the PD-specific enhancer element. So as you can see here, first we profile the epigenome by collecting over 50,000 the nuclei from the brain tissues. And then, as you can see here, we can cluster these cells based on epigenetic profiles, which reveals those 7 major the cell types. And then, using this information, we can clearly define the cell type specific epigenomes. So here you can see there are many cell type specific the enhancer regions from our data. And after that, by combining the bulk chips data, because to define enhancer regions, we need a specific histological mark. So by incorporating all the bulk chips data, we define PD specific the enhancers, which are dysregulating the brain tissue. And then by using the cell type specific epigenome, we can also annotate their response of the cell type. So as a result, as presented here, we have identified over 5,000 dysregulated enhancers in the PD brain tissues, and also defined and they are mostly cell type specific. Then what's the next? So after we define these dysregulated enhancers, the next step is we try to understand the association between these enhancers and the PDGR SNPs. So to this end, we first collect 3 different studies, and then by performing AD score regression analysis, we can see there are strong enrichment of the PDGR SNPs of these dysregulated enhancers in both neuronal and the non-neuronal cell types. So as demonstrated in the genetic variant presented in the essential locus in here, the patient carrying this variant present like a decreased like enhancer activities marked by these red bars compared to the enhancers in the normal like enhancers marked by the green bars in here. So as a control, we also compare to the SNPs obtained from the Alzheimer's disease and also other like neuronal like disorders. But here we cannot see such enrichment. So we can conclude that these enhancers are really specific and really they are highly associated with the PDGR SNPs. So by using the chromatin content information, we define their target genes. And as a result, we identify over 600 like the new both known and like, novel the target genes. So the number is actually largely expanded compared to like a previously known like a PD genes, which is less than 100. So we wanted to systematically investigate the function of like these genes by checking their expression profiles across the multiple individuals. So based on like the co-expression patterns, we can identify like 9 different like modular gene expression patterns present in here, and each gene set is marked by like a unique biological functions shown here. More importantly, these modularized target genes of the PDGR SNPs are not marked by not only marked by the distinct biological functions, but also they are dysregulated in a cell type specific manner. So, for example, if you look at the genes involved in like the first module, they are associated with response to unfolded protein or the RS metabolism. But as you can see here, these genes are specifically dysregulated in the microglia. However, if you look at the genes involved in like, the module 3, these genes are associated with negative regulation of the apoptosis, and their dysregulation pattern is specific to the oligodendrocyte and the astrocyte. So the main point of my study is, based on like a GR study, like based on genetic approaches, actually we can distinguish where these variants act their function in a specific cell type. But by using these single cell approaches, now we can precisely define their response of cell type to annotate their function. So to summarize my work, so today I gave this question in the beginning, the functional notation of the non-coding PDGR SNPs. And to address this, answer this question, I like to strongly propose the combined approach of the single cell omics and the 3D genome is very powerful to answer this question. So this work is led by my former grad student, Dr. Andrew Lee, and also the collaboration with Dr. Eliza from the NIA. So thank you for listening.
Thank you, Dr. Zhang. So Cindy, we're gonna have the question after the session. Thank you very much. Yeah. So that finally, we go to next speaker of this session, be Dr. Partha Majumdar. Actually, he's well-known the scientist in India, also in the world widely. So he is a National Science Chair of the Government of India. Also, he was the President of the Indian Academy of Sciences. Also, he served a lot of activity for the single cell organizing committee member in the HCA. So he will present us today. Yeah. Thank you.
Thank you, Yong-An. So I'm going to talk about single-cell transcriptomic analysis of oral cancer that has revealed 2 dominant cellular programs. This work I'm presenting on behalf of a whole team of people, the most important of which is she's sitting there. Her name is Celerine. So she's done most of this work that I'm presenting. This is a part of our work, continuation of our work from the International Cancer Genome Consortium that actually led to, naturally led to this single cell work. I'm going to talk about squamous cell carcinoma of the oral cavity, which is the 8th most common cancer. About 250,000 cases arise annually, about 2/3 of them in developing countries, and about 128,000 deaths happen annually. It accounts for about 1/3 of all tobacco-related cancers in India. This is a cancer type that's most prevalent in India among the males and is also very highly prevalent in Southeast Asia. The site distribution of oral cancer is very interesting in the sense that in India, one finds that tongue and floor of mouth occupies about 25% of the cases. The remaining 75% of the cases are rest of the oral cavity, excluding the tongue. In the West, what one finds is an exact contrast, where you find the tongue cancers to be the most prevalent. About 70% of the cases are tongue cancer. So this gives us an opportunity to look at what happens in gingivobuccal oral cancer and contrast it with tongue cancer. Essentially, tobacco chewing is the most predominant form of environmental insult in the oral cavity in India and in Southeast Asia. In the West, it's mostly in the form of tobacco smoking. Our information on patients, this is about 500 patients that we looked at. Most of them were male, most of them were middle-aged. Most of the patients actually present very late. They were exposed to tobacco. Almost all of them were exposed to tobacco, and HPV infection was nearly absent. Our work as a part of the International Cancer Genome Consortium revealed that tumor suppressors are the main driver genes, and we identified 10 driver genes, which all work as tumor suppressors, except for 1 or 2. Most of them work as tumor suppressors. If we look at the distribution of tumor suppressors and look at the distribution of tumor suppressors among patients, what one finds is that the patients can be subgrouped. The major subgroup is driven by TP53. The other major subgroup is driven by FAT1 and caspase-8. Of course, you can find smaller subgroups, and these subgroups essentially differ in terms of their survival probabilities after treatment. Don't have the time to get into the details of it. The recurrence frequency distribution, if one looks at recurrence frequency distribution among these 500 patients, one finds a very long tail. Of course, not all of them recur very, very frequently, but this really tells us that there is a lot of heterogeneity, and this, in fact, led us to the single-cell analysis in a natural way. Aside from this, I must also say that the long recurrence tail distribution led us to certain pathways, in particular the arachidonic acid metabolism pathway that might actually impact on how one treats these kinds of cancers. But again, like I said, that I don't have the time to get into that. The objectives of this project, of the single-cell project, which was a natural consequence of our bulk sequencing projects under the International Cancer Genome Consortium, which started to profile cell type diversity in this cancer type and identify gene expression programs in different cell types, and to also understand, to investigate cellular interactions that happen within the tumor ecosystem. We profiled 12 patients. Most of these patients, all of these patients were gingivobuccal oral cancer patients. Some of them had a precancerous lesion that turned into malignancy. The precancerous lesion is known as oral submucous fibrosis. Most of them, like I said, were of a high grade. In terms of classification of malignant and non-malignant cells, we used INFER-CNV, but we also devised our own method. This is particularly interesting because gingivobuccal oral cancer is an epithelial cancer. To identify malignant cells, what we did was to identify a reference expression profile of non-epithelial cells and try to find cells that are most distant from that reference sequence. And every one of the patients had this anti-mode, which is what we had expected. Don't have the time to get into the details of this methodology, but essentially it worked very well. As a matter of fact, it worked better than INFER-CNV in the sense that the hallmarks of cancer genes were more enriched in this particular method of identification of malignant cells than INFER-CNV. We call this method as a distance profiling method. If you look at the profile of the OSCC, the gingivobuccal oral cancer patients, of course, there is a lot of variation. There are 7 cell types, and these cell types are not uniform across patients. So those are the patients, and essentially what one sees is that there is a difference in distribution of these different cell types among the patients. Each cell type has representative cells from all of the samples. If you look at the malignant cells, and there were about 7,500 malignant cells, we can cluster them by cell type or we can cluster them by patients, but it turned out that malignant cells most of the time cluster by patient. And the reason why it clusters by patients, we think, This has also been found in other kinds of cancers where malignant cells cluster by patients, essentially because the somatic alterations are quite different from one patient to another, and that may be the reason why it's clustering by patients. Non-malignant cells don't show that feature. We found that there is an enormous role of EMT, epithelial to mesenchymal transition. About 41% of the malignant cells express genes that are involved in EMT. Some of these genes were also involved in TGF-beta signaling, Notch signaling, and P53 signaling pathways that probably drive these and regulate the EMT in these particular in these cells. We also found that HIF1A expression level positively correlates with expression of these EMT-driven cells, and that's probably a hallmark of partial EMT. So we believe that EMT and partial EMT that actually comprises or actually impacts on about 41% of the malignant cells is a major dominant cellular program. There's also fetal and germline epithelial cells that are abundant among the malignant cells. About 40% of the malignant cells comprise fetal and germline epithelial cells. Again, we looked at various signature sets, and we found that major clusters of cells in these patients actually provide signatures of these fetal epithelial cell types. There are enriched expressions of cancer testis gene family, including various kinds of cancers, and those were also observed in about 30% of the malignant cells that are indicative of germline epithelial cells. And about 32% of them, and these are not disjoint classes. I mean, there's obviously overlap. Otherwise, the total sum of these percentages will exceed 100. So about 32% of the malignant cells express genes of the immune-related pathways, as one can see. And these immune-related pathways are quite interesting. The cells that are enriched for expression of genes on pathways are antigen-presenting and processing and presentation genes, NK cell-mediated cytotoxicity genes, cytosolic DNA sensing genes, and so on. So these various pathways are quite important in driving cancer and in cancer progression. And these immune-related pathways turned out to be important in about 32% of the malignant cells. T cells are abundant in tumors with diverse subtypes and levels of infiltration. They accounted for about 36% of all cells in the tumors. The T cell population was transcriptionally heterogeneous. The major cell clusters comprised regulatory T cells, CD4 positive T cells, CD8 positive T cells, and double negative T cells and NKT cells. So there is a lot of diversity. Even though T cells are abundant, there are various subclasses of T cells that are enriched in this particular cancer type. Gingivobuccal oral cancer, which is, like I said, the most predominant form of cancer among males in Southeast Asia, exhibits large intra-tumor cell heterogeneity. The fetal cellular reprogramming and EMT, partial EMT play, these are the dominant programs that play major roles. And that's where our work has gotten to in this short time. That's all I could present, but there are many other features, many other interesting features that might actually prompt us to undertake other kinds of research on oral cancer. But again, like I said, that this is a short presentation. Don't have very much of time. Thank you very much.
Thank you, Dr. Okay, so we are going to close the session because they're going to be followed by panel discussion. Thank you very much for your participation.